I'm going to talk about everything you need to know for proteins and amino acids. So amino acids are the building blocks of proteins. The structure of an amino acid is a central carbon, a hydrogen, an R group, a carboxyl group, and an amino group. If you take the amino group off of an amino acid, it becomes an alpha keto acid, also called a carbon skeleton. Some of our amino acids are essential, which means we need to get them from diet. The way to remember this is Private Tim Hall. So phenylalanine, valine, threonine, tryptophan, isoleucine, methionine, histidine, arginine, lysine, leucine. Arginine is essential for infants, so infants need to get arginine through their diet. Arginine becomes non-essential for adults, so we end up making arginine from scratch as an adult. Proteins are first broken down mechanically through chewing in the mouth. They get broken down into a little bit smaller polypeptides, and then breakdown starts in the stomach and then finishes in the intestine. HCl is stomach acid that turns this proenzyme pepsinogen into pepsin. This will break down proteins a little bit smaller in the stomach, and then those proteins will continue down into the intestine. The intestine will secrete peptide hormones known as secretin and CCK. And secretin will tell the pancreas to release bicarbonate. CCK will tell the gallbladder to release bile into the intestine to emulsify lipids, and it'll tell the pancreas to release a bunch of proenzymes like trypsinogen, chymotrypsinogen, and procarboxypeptidase. So the first proenzyme, trypsinogen, is activated into its active form by enteropeptidase. So enteropeptidase turns trypsinogen into trypsin. Then trypsin activates other proenzymes. So trypsin activates chymotrypsinogen to turn into chymotrypsin, and procarboxypeptidase to turn into carboxypeptidase. And by the end of all of these reactions, proteins should be broken down into single amino acids, dipeptides, so 2 amino acids long, and tripeptides, 3 amino acids long. And we break them down into single amino acids, dipeptides, and tripeptides so that they're small enough to be absorbed. And we absorb these amino acids for 3 reasons. We synthesize new proteins from those amino acids. We make new amino acids, or we break the amino acids down even further to use the carbon skeletons for energy. There's a disease process known as Hartnup's disease, or neutral hyperaminoaciduria. It's caused by a defective neutral amino acid transporter. Tryptophan is a neutral amino acid that's affected by this defective transporter. So if tryptophan is not absorbed, it'll be excreted in the urine, and that's why you have neutral hyperaminoaciduria, because you have a bunch of neutral tryptophan in the urine. Tryptophan is a precursor for B3. If we don't have tryptophan, we can't make B3, and when you don't have B3, you end up having pellagra. The way to treat Hartnup's disease is by treating with B3. Some definitions that relate to proteins and amino acids. Essential amino acids, like we said, are those that you need from diet. There's 10 of them in that private Tim Hall mnemonic. And then non-essential means that your body can synthesize these from scratch. Conditionally essential means that because of a disease condition, a non-essential amino acid now becomes an essential amino acid, because if we have a disease where we can't make an amino acid that we could normally make, we have to get it from diet. The normal protein intake is 0.8 grams per kilogram of body weight. I wrote true or false. Proteins are stored. That's false. We never store proteins. We're constantly breaking them down into amino acids and then putting amino acids together to synthesize proteins based on our energy requirements. There's also complete and incomplete proteins. Complete proteins means it has all the essential amino acids. Incomplete means that it's lacking one or more essential amino acids, and that will lead to a limited amount of proteins that are made. So if you're missing lysine, for example, one of the essential amino acids in the private Tim Hall mnemonic, every protein that requires lysine won't be able to be made. Plants lack 3 amino acids: methionine, lysine, and tryptophan. So I remember it as me like tripping, and I say on shrooms because shrooms are a plant. So me like tripping. Just help me remember it. So that means essentially if you eat a plant-based diet, you'll probably need to take a supplement that includes these 3 amino acids that you're not getting because you're not eating meat. There's 2 diseases associated with proteins: kwashiorkors and marasmas. Kwashiorkor is when you're not getting enough protein, but you're getting enough carbs and fat, so you have enough energy. You'll have edema because, so you'll have like this picture demonstrates kwashiorkor. So they'll have skinny limbs and then a protruding stomach due to edema, because if you lack proteins in your blood, there will be more proteins in your tissues. So water follows solute. Water will go into your tissues and then cause edema or swelling. Marasmas is when you don't have enough protein, carb, or fat intake, so you have no energy, and it's known as wasting or emaciation. Moving on, transamination is a process where you transfer an amino group from one amino acid to a keto acid, or from one amino acid to another amino acid. So it's just transferring one amino group. And this occurs in all tissues, but mainly the liver. So here's an example of that. So you take any amino acid, and this triangle signifies the amino group. So you take an amino acid, you transfer its amino group onto alpha-ketoglutarate, which is an alpha-keto acid. When alpha-ketoglutarate receives an amino group, it becomes glutamate. And then this original amino acid will, when it loses its amino group, it'll become its corresponding alpha-keto acid. And transamination reactions always use transaminase enzymes, and they require pyridoxal phosphate or B6. You can also go the other way around, where glutamate donates an amino group. So glutamate has the amino group. It puts it onto a keto acid. That keto acid will become its corresponding amino acid because it gained an amino group. and then glutamate will turn into its pair alpha-ketoglutarate as a result. This is just a little fact, but lysine is the only amino acid that does not have an aminotransferase enzyme. So that's just something to remember. There are certain pairs to remember. There's amino acid and alpha keto acid pairs. So like we just saw, glutamate is always paired with alpha ketoglutarate. And this reaction is reversible. This goes back and forth, and glutamate dehydrogenase can either turn glutamate into alpha ketoglutarate or alpha ketoglutarate into glutamate. Alanine and pyruvate are pairs as well, and this transaminase reaction or transamination reaction is done by alanine aminotransferase, ALT for short. Aspartate and oxaloacetate are always paired, and this reaction is done by aspartate aminotransferase. These all require B6 as cofactors, and an increase in serum ALT and AST, these liver enzymes, indicates that you have liver damage. Another reaction done with amino acids is known as oxidative deamination. This is the process of removing an amino group for excretion, and it occurs in the liver or kidney. Glutamate is always used for deamination reactions. So glutamate dehydrogenase turns glutamate into alpha ketoglutarate, and in the process, glutamate removes its ammonia group. Ammonia is toxic in our blood, and I'll explain why a little bit later, but we don't want ammonia in our blood. So if oxidative deamination is occurring in the liver, we'll turn ammonia, put it through the urea cycle, and turn it into urea, so that now urea can travel through the blood because it's not toxic. Urea can travel through the blood, go to the kidney, and just be excreted. If oxidative deamination is occurring in the kidney, we don't need to convert ammonia to urea. We can just excrete urea out of we could just excrete ammonia out of the body. Now there's two other reactions known as transamidation with a D and deamidation with a D. So transamination is transferring a second amino group from asparagine or glutamine to an alpha keto acid or an amino acid. So the D stands for double in my mind, because we have deamination and deamidation. Deamidation and deamination. They both involve removing an amino group, but deamination with an N is involved with removing one amino group. Deamidation with a D is involved with removing a second amino group to be excreted. Transamination versus transamidation. So transamination with an N is transferring one amino group. Transamidation is transferring the second amino group. I hope that makes sense. There's something called the alanine cycle, where we essentially, alanine is able to travel through the blood to bring ammonia from muscle to the liver. Because in our muscle, we have a bunch of protein, and when protein is broken down, it creates nitrogenous waste. So we put that nitrogenous waste into alanine so that alanine can travel through the blood and be a non-toxic way to get ammonia out of the body. So it can travel to the liver where it could go through the urea cycle and turn into urea. So it'll make a little more sense when I explain this. So you start with any amino acid with its amino group. You transfer that amino group onto alpha-ketoglutarate. Alpha-ketoglutarate becomes glutamate, and the amino acid becomes its corresponding keto acid. This is a transamination reaction, so it requires B6. Once we get glutamate, we need to get alanine, so alanine could travel to the liver. So we do another transamination reaction where glutamate puts its amino group onto pyruvate. Pyruvate turns into alanine, and glutamate turns into alpha-ketoglutarate once again. ALT does this reaction, alanine aminotransferase. And alanine now can travel through the blood to the liver, where it'll do a transamination reaction and turn back into pyruvate. So alanine will put its amino group on alpha-ketoglutarate to turn into pyruvate. Alpha-ketoglutarate becomes glutamate. It becomes glutamate because now we could do a deamination reaction to remove ammonia. So glutamate turns into alpha-ketoglutarate, removes ammonia. This is done using glutamate dehydrogenase and B3. I think I forgot to mention that oxidative deamination requires B3. So ammonia goes through the urea cycle, turns into urea, so now urea can go to the kidneys to be excreted. This is just a little fact here that always came up, is that alanine and glutamine are highest after 2 days of fasting. And that makes sense because after 2 days of fasting, we're breaking down protein stores. And when you break down protein, you produce nitrogenous waste like ammonia. And we put ammonia inside of alanine and glutamine so that they can travel freely through the blood to either the liver or kidney so that the ammonia could be removed and be excreted. 3 enzymes that bind up free ammonia. This came up a lot too. So glutamate dehydrogenase is the enzyme that is seen here. So we put ammonia and alpha-ketoglutarate together to get glutamate. So glutamate is carrying an ammonia group within it, so it's binding up free ammonia essentially. Glutamine synthetase is another enzyme that will bind up free ammonia. When you turn glutamate into glutamine, you put ammonia in with glutamate to make glutamine, so that's binding up free ammonia. And then CPS1 is seen in the urea cycle, so it's incorporating ammonia and either carbon dioxide or bicarbonate, binding up ammonia to get carbamoyl phosphate. But now on to the glutamine cycle. So glutamine is similar to alanine in that it transports ammonia, but glutamine is even more prevalent than alanine because alanine just transports ammonia from muscles, but glutamine transports ammonia from all cells to the liver or kidneys. So same thing happens where you have any amino acid putting its amino group on alpha-ketoglutarate so that we can get glutamate. Glutamate has one amino group. Glutamine has 2 amino groups, so we need to add ammonia and glutamate together using glutamine synthase to get glutamine. Once we have glutamine, it could travel to either the liver or kidney. So let's just talk about it in the liver. So glutamine's in the liver. We want to turn it back into glutamate. So we remove one of its amino groups. In this amino group, in the form of ammonia, will go to the urea cycle, turn into urea so that it could go to the kidney. We're left with one amino group on glutamate. Glutamate will use glutamate dehydrogenase to turn into ammonia and alpha-ketoglutarate. This ammonia will do the same as this one. It'll go through the urea cycle, turn into urea, and then travel to the blood and go to the kidney. This reaction is a deamidation reaction because we are removing an ammonia or an amino group for excretion. This is deamination reaction because we're removing one amino group. This was removing glutamine's second amino group, and then this is removing glutamate's amino group. So same thing happens in the kidneys. The only difference is when you remove ammonia, it could just be excreted. It doesn't have to go to the urea cycle because you're already at the kidneys. The kidneys can just excrete ammonia as it pleases. Something to note is that when you have super high nitrogen levels in your urine, the basicity of the ammonia can give you a urinary tract infection because urine is typically on the more acidic side. So earlier I mentioned that ammonia is toxic. That's why we turn it into urea, because urea can travel through the blood and bring ammonia to the liver or kidney. So if we have ammonia in the blood for some reason, it will increase the blood pH because this is a basic molecule. It can cross the blood-brain barrier and kill astrocytes. And astrocytes are a type of neuroglia that protect the nerves in the blood-brain barrier. And ammonia inhibits the TCA cycle. So now the urea cycle, this process occurs in the liver. You're turning ammonia into urea. There's no energy produced from this pathway. So here are the steps. So you either take CO2 or bicarbonate, a carbon source. You combine it with ammonia using carbamoyl phosphate synthetase 1, CPS1, to make carbamoyl phosphate. And NAG is the cofactor that stimulates CPS1. Just remember that. Carbamoyl phosphate and ornithine turn into citrulline using trans-ornithine carbamoylase. Citrulline combines with aspartate to form arginosuccinate using arginosuccinate synthetase. Arginosuccinate lyase cuts arginosuccinate into arginine and fumarate. Arginase cuts arginine into urea and ornithine, and then ornithine is reused. This pathway has a lot of diseases. If you inhibit CPS1, you'll get a buildup of ammonia, which is known as hyperammonemia 1. If you inhibit trans-ornithine carbamoylase, the same thing will happen. You'll have a buildup of ammonia because you'll have a buildup of carbamoyl phosphate, but then it'll also build up and inhibit what came before it. So you'll have hyperammonemia 2, just caused by a different enzyme. If you inhibit arginosuccinate synthetase, you'll have a buildup of citrulline, which is known as citrullinemia, and that's in the blood. If you inhibit arginosuccinate lyase, you'll have arginosuccinic aciduria. This is the only one in the urea cycle that's causing a problem, a buildup of a molecule in the urine, because the rest of these are occurring in the blood. If you inhibit arginase, you'll have a buildup of arginine, so you'll have hyperargininemia. You can treat hyperargininemia by a low-protein diet, because if you have a high-protein diet, this pathway is going to be occurring, because when we eat protein and we break it down into amino acids, we produce ammonia, and that's a nitrogenous waste. We don't like having ammonia in the blood, so we gotta convert it to urea. So the less protein we have, the less urea we're gonna make. And over here, it's worth mentioning that when fumarate's produced, it'll go through the TCA cycle to become oxaloacetate. And then, as we mentioned earlier, oxaloacetate and aspartate are pairs, so they can do a transamination reaction. Glutamate can put its amino group onto OAA, and then OAA can turn into aspartate, and then it could be recycled. So there's a lot of recycling going on here. But once we get urea, since the urea cycle happens in the liver, we have to send the urea through the blood, to the kidneys, so it could be excreted. And insulin decreases urea cycle. Urea cycle is always going, but insulin decreases urea production because insulin promotes storage, and this cycle is happening because of protein breakdown. And we only have protein breakdown when we're fasting or starving, so the fed state will inhibit urea a little bit more, urea cycle. The next thing is carbon skeleton. So proteins are broken down to amino acids, and then amino acids contain an amino group and the rest of its structure, an alpha keto acid, otherwise known as a carbon skeleton. And these carbon skeletons can be broken down further to become intermediates in different pathways. Glucogenic means that the carbon skeleton can be used to produce glucose. Ketogenic means the skeleton will be used to make ketones or fatty acids. And glucogenic and ketogenic mean they do both. So the only strictly ketogenic amino acids are lysine and leucine. Glucogenic and ketogenic are, I remember, PIT with 3 T's: phenylalanine, isoleucine, tryptophan, threonine, tyrosine. And then that leaves glucogenic amino acids to be all others besides these. So there's a lot of little memory tricks I used for this. So pyruvate, a bunch of these amino acids could be broken down into pyruvate, so that pyruvate can turn into oxaloacetate and go through gluconeogenesis to produce glucose. So I remember 3 cycloal's seriously trip gleefully: threonine, cysteine, alanine, serine, tryptophan, glycine. For oxaloacetate, I always shorten it as OAA. The 2 A's stand for asparagine and aspartate. Acetyl-CoA can be used to produce ketones, so that's why these are ketogenic. So it's LILIT with 2 L's and 2 T's. So lysine, leucine, isoleucine, tryptophan, threonine. This is Pretty Little Tires is acetyl-CoA. Phenylalanine, leucine, tyrosine. This is ARG, his proglutes for alpha-ketoglutarate. So arginine, histidine, proline, glutamine, they all can turn into glutamate. And then glutamate turns into alpha-ketoglutarate, as we know. It sucks to vomit for succinyl-CoA. So valine, methionine, isoleucine, threonine. I remember you got a fatty with a pH. So phenylalanine, aspartate, tyrosine for fumarate. What else did I want to mention? Oh, there's threonines here 3 times here, here, and here. Tyrosine's mentioned twice. Tryptophan's mentioned twice. So just, there's a lot of T's, so make sure you know which one's which. The protein or the amino acid catabolism pathways were super confusing, but I just focused on the steps where there were something wrong with like the disease steps essentially. So this is seen in the methionine, threonine, BCAA pathway where propionyl-CoA uses propionyl-CoA carboxylase to turn into methylmalonyl-CoA. Then methylmalonyl-CoA mutase turns methylmalonyl-CoA into succinyl-CoA. If you inhibit propionyl-CoA carboxylase, propionyl-CoA will build up, and that's known as propionic acidemia. If you inhibit methylmalonyl-CoA mutase, you'll have a buildup of methylmalonyl-CoA, so it'll be known as methylmalonic acidemia. I think those can also be acidurias. When it comes to BCAAs, branched chain amino acids, it's I love valine, isoleucine, leucine, and valine. They all use an enzyme known as branched chain amino acid dehydrogenase. And dehydrogenase, this one uses B1, 2, 3, and 5 as cofactors. If you inhibit BCAD, you'll have a buildup of isoleucine, leucine, and valine, and those will cause maple syrup urine disease. You'll see a buildup of them in your urine, and it'll cause them to smell sweet. It'll cause your urine to smell sweet. The methionine pathway is important. Methionine turns into SAM, and she wants you to remember that it's a methyl donor. SAM is an important methyl donor. SAM turns into SA, and then you get homocysteine. Homocysteine and serine combine to form cystathione. Cystathione turns into cysteine. When you put glutamate, glycine, and cysteine together, you get glutathione. If you inhibit cystathione synthase, the enzyme that turns homocysteine into cystathione, you'll get a buildup of homocysteine in the urine and the blood. So homocysteine, urea or emia, that'll cause an increased risk of blood clots and cardiovascular events. We don't want that. So we need B3 to help cystathione. We also need B9, B12 so that we can turn homocysteine back into methionine. There's another pathway for tryptophan where tryptophan produces serotonin, and serotonin turns to melatonin, so I always remember that. On Thanksgiving, you eat turkey because tryptophan's high in turkey. That'll make you happy to see your family, but then you get really tired on Thanksgiving and want everybody to leave. Tryptophan also turns into kynurenine and then anthralinate. And anthralinate uses alpha-ketoadipic dehydrogenase and glutaryl-CoA dehydrogenase to turn into acetyl-CoA or B3. So that's why tryptophan is a precursor to B3. If you don't have tryptophan, because of Hartnup disease, you can't make B3. So then that's why you need to supplement with B3. But if you inhibit these enzymes, you'll end up with alpha-ketoadipic aciduria or glutaric aciduria. And phenylalanine is an important pathway. Phenylalanine hydroxylase turns phenylalanine into tyrosine. Tyrosinase turns tyrosine into L-DOPA. L-DOPA turns into dopamine. Dopamine turns into melanin or norepinephrine, and then norepinephrine turns into epinephrine. If you Oh, and then tyrosine also breaks down to form fumarate or acetoacetate using homogentisate oxidase. So if you inhibit phenylalanine hydroxylase with its cofactor biopterin, then you can get something known as phenylalanine ketonuria, where phenylalanine builds up and it causes a musty odor, and you'll have fair skin and hair. If you don't have enough vitamin C or you inhibit tyrosinase, tyrosine can't go on to produce melanin. That's the pigment in your skin and your hair. And if you can't produce melanin, then you might be albino. If you inhibit homogentisate oxidase, which uses vitamin C, you'll have something known as alkaptonuria, which is dark urine when exposed to air. The next little thing is Williams disease is caused by inhibiting phosphatase, and that's the enzyme that turns 3-phosphoserine into serine. So I remember Serena Williams for this disease. Williams disease is when you can't make serine. Serena Williams. Glycine. I remember its special products for Create Me Pure Glutes. So glycine can make creatine with arginine. Glycine plus succinyl-CoA equals heme. So that's the me. Pure is purines. Glycine, glutamine, aspartate. And then glutes is glutathione, which is what we saw here. Glycine, glutamate, cysteine. And if you break down glycine using the glycine cleavage system, you'll get CO2 and an amino group. And if you block the glycine cleavage system, you'll get a buildup of glycine. That's known as non-ketotic hyperglycemia. I think that's pretty much all the important pathways. Yeah. Hi. Sorry. This pathway is crazy. Just remember that arginine can form nitric oxide and creatine when paired with glycine. Arginine uses arginase to turn into ornithine. We saw that in the urea cycle. Ornithine is used to produce polyamines like spermidine and spermine. Glutamate can turn into glutamine. Glutamate can turn into alpha-ketoglutarate. Glutamate can turn into GABA through glutamate decarboxylase and B6. If you inhibit, or if you don't have GABA, that can cause abnormal muscle tone. GABA is an inhibitory neurotransmitter, and it helps inhibit. Oh my god, I can't talk. It inhibits like non-coordinated muscle movements. So you need GABA to help you not make random muscle movements. Histidine does a decarboxylation enzyme to become histamine, and decarboxylation enzymes, same, are B6, and histidine can become glutamate using histidinase and B9. What else do I have? Amino acids produce special products. We talked about the glycine ones, the Create Me Pure Glutes. If you don't have heme, you can become photosensitive. So that means your skin can be sensitive to the sun. Catecholamines come from tyrosine. I remember when you're tired, you need to wake up. So catecholamines, norepinephrine, and epinephrine will wake you up. We just saw GABA comes from the decarboxylation of glutamate. I remember that as your glutamate, listening to ABBA, because Mamma Mia. We just saw histamine comes from decarboxylation of histidine. Those kind of go together. Arginine forms nitric oxide. I just remember that as when you argue, it's because someone told you no. Polyamines come from ornithine. We saw that, the spermine and spermidine. Tryptophan forms serotonin and melatonin. Remember, when you eat turkey, you get happy and sleepy. And then carnosine is from histidine and alanine. And I remember that is histidiner is a la carte. Histidiner a la carte. It's kind of stupid, but it helped me. I think the last thing is plasma proteins. So all plasma proteins are the proteins that are in our blood plasma. They're all made in the liver except gamma globulins. And gamma globulins are known as immunoglobulins or antibodies. And we know from immunology that our antibodies are produced from plasma B cells. All plasma proteins are glycoproteins, so they all have a glucose or a sugar on their surface, except albumin. Albumin has no sugar on its surface, it's just a plain old protein. There's alpha-1 globulins, alpha-2 globulins, beta globulins, immunoglobulins, and albumin. So alpha-1 globulins, they both start with alpha, which is helpful. Alpha-1 antitrypsin. Antitrypsin, it inhibits trypsin, which is a protease that destroys lung tissue. So antitrypsin helps inhibit trypsin so that it doesn't destroy lung tissue. If you don't have antitrypsin, you may destroy your lung tissue and end up with emphysema. Alpha-fetoprotein, or AFP, it has feto, so I remember that as fetus. It's a protein that protects the fetus from the mom's immune system, because we learned in immunology that the mom thinks the baby's a virus and wants to attack it. But this protein prevents that from happening. Alpha-2 globulins, there's two alpha-2s, ceruloplasmin and haptoglobulin. Ceruloplasmin transports copper. Haptoglobin transports hemoglobin. You have to grab onto hemoglobin. That's how I remember that. If you have low ceruloplasmin, you won't be able to transport copper, so it'll build up in the tissue and it'll cause Wilson's disease. There's 4 beta globulins. Transferrin transports, so ferritin, you can think of iron, it transports iron. Fibrinogen helps blood clotting, form a fibrin clot. CRP, we learned in immunology, is the marker for bacterial infection. It starts the complement cascade to kill bacteria. Beta-2 microglobulin, you can remember it's a beta globulin because it starts with a beta. And then it's just part of the MHC HLA protein that is a marker for self. Immunoglobulins are antibodies, IgG, IgM, IgA, IgD, IgE. We know from immunology that they bind to antigens during the immune response. Albumin is like the most important one that's always talked about. It's a simple protein, like we said. There's no sugar on its surface, it's just a protein. It's important for transporting a lot of molecules. So it transports B, C, D, and F. So kind of in alphabetical order. Bilirubin, calcium, drugs, and fatty acids. And it maintains oncotic pressure in the blood. So albumin, oncotic pressure is the idea that in our blood, if you have plasma proteins, water follows solute. So the plasma proteins in our blood will bring water back in to the blood from our tissues. If we have low albumin, hypoalbuminemia, if we have low albumin in our blood, there will be more albumin or more solutes in our tissues, which will cause fluid to move out of the blood and into the tissues, and then that will cause edema. During acute phase inflammation or an infection, we make a few of these plasma proteins and we inhibit the production of a few of them. So I remember that we're making 1 alpha 1, 2 alpha 2s, and 2 betas. So you make 1 alpha 1, that's the antitrypsin. You make, and this will inhibit proteases. You'll transport copper. You'll transport hemoglobin. You'll stimulate blood clots. And since it's an infection, you'll make more CRP to induce the complement cascade. We make less TAP, transferrin, albumin, and prealbumin. That's how I remembered that. And that's all of proteins and amino acids that I can think of.